So our first speaker today is Dr. Megan Rowland. She did her undergrad at the Vancouver Island University before moving to Western University in Ontario for her PhD with Natalie Baribeau and Frank Bier, studying premature aging and myelination in ATRX mutant mice. For her postdoc, Megan moved back to the West Coast, the University of British Columbia, to work with Annie Cernia, where she holds a prestigious Michael Smith postdoc fellowship. Her postdoc work focuses on understanding how perturbation of gene regulatory networks can lead to neurodevelopment disorders, in particular, how chromatin remodelers influence brain development. And I'm very excited to hear her talk today, titled Investigating the Role of Baf53b in Neuron Development and Autism Spectrum Disorder. So please join me in giving a warm, albeit virtual, welcome to today's first speaker, Dr. Megan Rowland.
Great, thank you so much for having me. I'm really excited to tell you a little bit about part of my project, which is looking at this role of Baf53b in neuron development and autism spectrum disorder. So just a quick overview of autism spectrum disorder. It's a really prevalent neurodevelopmental disorder, and those with ASD have restricted interests, repetitive behaviors, and difficulties with social communication. And while it's well established that it is heritable, it's complex. There's hundreds of genes that have been implicated. And this heterogeneity has really led to a goal in the community to identify common pathways and targets that are disrupted in ASD, with the hope to understand this disorder better and potentially develop therapeutics. Now, there are two categories that some of these genes tend to fall into that are implicated in ASD. One of these is those involved in synapse development. So those are the ways that neurons communicate with one another. And the other is chromatin remodelers. And so I'm sure I don't have to describe this to this audience, but there's a large amount of DNA that needs to be compacted into a very tiny nucleus. And at the same time, this DNA needs to be accessible for gene expression. And so that's where chromatin remodelers come in. I study switch SNF, or as in mammals, we like to call it the BAF complex, BRG1 associated factor complex. And this complex uses the energy of ATP to either slide or eject nucleosomes to either increase space or to close down that chromatin, usually to increase or decrease gene expression. And so this is a large multi-subunit complex, and the subunits can actually combine in different ways to allow for cell type specificity. And a really classic example of this is during neuron development, where we have a switch from the neuroprogenitor BAF complex to the neuronal BAF complex upon differentiation. And so this is the neuroprogenitor BAF complex, which is defined by the subunit shown, as well as BRG1, which is the ATPase in the complex. And when this complex is present in the cell, it promotes proliferation of neural stem cells and suppresses differentiation. Upon differentiation, the subunits exchange for the one shown in the bottom purple complex, and then we have the neuronal BAF complex. This is the complex that I study. And when this complex is present, it's important for gene expression involved in neuron differentiation and morphogenesis. And so my favorite protein is Baf53b. It's close to BRG1, they interact, and I will just note that its non-neuronal homologue, Baf53a, is really similar to it. They differ only in a small region called subdomain 2. So we think that this area is really important for its neuronal specific function. And so there's been some work on Baf53b, mostly in excitatory neurons. So there's sort of two big classes of neurons, either excitatory or inhibitory. You can think about it like the gas and the brake in the brain. And most of this research thus far has been in excitatory neurons. And it seems like Baf53b is important for dendritic outgrowth and branching. So this is where the neuron receives inputs from other neurons, as well as synapse formation, once again, how the neurons talk to each other, and some functional things like social memory, which is highly relevant to ASD, as well as long-term memory and synaptic plasticity, which is also relevant to ASD, as patients often also exhibit intellectual disability. And I think most importantly, very recently, Baf53b variants have also been found in individuals with ASD. And so here's just a schematic of both the Baf53b gene and the Baf53b protein, showing the discovered variants in individuals with ASD. You can see that they span the gene, they don't concentrate in a single area, and most of them, and the ones shown in black, are homozygous recessive. There are a couple dominant mutations, those are the ones shown in red. And I will also note there is one mutation in that subdomain 2, the divergent region, that we're really interested in exploring its function and how it affects the Baf53b protein. So that leads me to my hypothesis, which is deletion or mutation of Baf53b will alter gene expression that's required for neuron development, resulting in abnormal dendrite growth and ASD-relevant behaviors. So I'm just going to focus on a small portion of this hypothesis today. But first, what we really wanted to do was confirm that Baf53b is expressed in both excitatory and inhibitory neurons, since we're interested in both populations. And so we took some RNA sequencing data from cell types brain map and found that there is enrichment of Baf53b, shown in blue, in both GABAergic inhibitory neurons and glutamatergic excitatory neurons. And this is seen both in the human and the mouse cortex. So that was great confirmation for us that it's neuron specific and specific in those inhibitory neurons as well. And so to study its function in these two cell types, I wanted to establish a mouse primary neuron culture system. And to do this, I can actually isolate both excitatory and inhibitory neurons from the same mouse brains. So I isolate the cortex to get excitatory neurons and the ganglionic eminences to get the inhibitory neurons at embryonic day 14.5. And we use the Crelox-P system to delete Baf53b. So these mice are double floxed for Baf53b. I culture the neurons. And then at 1 day in vitro, I infect the neurons with an adeno-associated virus, either carrying the Cre recombinase or not. Those that get the Cre recombinase will excise whatever's between those LOX-P sites, and then there is no protein product that's produced. And then at 7 days in vitro, I process the neurons for either immunofluorescence staining to look at neuron branching, or for RNA sequencing or RNA work. So just a quick overview of the conditions that we have. So these are the plasmids that the AAVs actually deliver to the neurons. This is our control plasmid. We just have a TD tomato reporter driven by the SYN1 promoter, which is neuron specific. So we can see which neurons actually get the plasmids. So in this case, we get a red neuron that still has the Baf53b protein. And then in our knockout condition, we have the same plasmid, but instead this plasmid has Cre. And so we get a red neuron without Baf53b. And what I found in the excitatory neurons after staining for the TD tomato, you can see in the control condition on the left, a very nicely branched excitatory neuron that still has Baf53b. And then when Baf53b is deleted in the image on the right, you can see a very stunted neuron. And we can actually quantify this by something called show analysis. And this is just a program that I use to draw concentric circles around the soma of the neuron. And then every time one of those circles crosses a branch, it gets counted. So you end up with a graph that looks like this, where it's number of intersections plotted against the distance of the soma. And in the control condition, lots of branching closer to the soma, which sort of tapers off as you get further away from the soma, and a significant decrease in that in our knockout condition. I then wanted to look at what might be going on at the transcriptional level in these excitatory neuron cultures. And I found something a little bit interesting. First of all, Baf53b was downregulated, which was great. And when I looked at the Go terms, which SNF superfamily type complex was enriched in the decreased condition. But I also found that there was an increase in some inhibitory neuron genes. And this was really unusual because these were excitatory neurons, or they were supposed to turn into excitatory neurons. So this sort of pointed to an area where there might be a misregulation of differentiation or an alteration to the development of these neurons. I then wanted to look at inhibitory neurons specifically, and this is some work that was led by Anne-Marie, a master's student in the lab. And so we've cultured these inhibitory neurons and done sort of the same thing as I showed with the excitatory neurons, where we get this nicely branched control neuron and sort of stunted, reduced branching in the knockout neurons. And you can see that that's significant as well. So the next part of this that I'm really excited about is actually looking at these variants that are found in ASD. So here are just the variants that are found in those with both ASD and intellectual disability. And I've created plasmids that have 2 of these mutations. So the first is the C230A to G, which is once again in that subdomain 2, the divergent region, as well as the C1177G to A mutation, which is found in the C terminus of the protein and is thought to mediate the protein-protein interactions between Baf53b and BRG1. And just a quick overview once again of our plasmid setup. So we can actually replace the endogenous Baf53b with these variants. And we use, again, the CreLox-T system. So I will use the adeno-associated virus to infect the neurons with both the Cre plasmid, as I described earlier, as well as this flex on Baf53b wild type plasmid. This is our control condition. And this is a really nice system where we get, we have the Baf53b gene with a GFP reporter in the reverse orientation. And this actually flips in the presence of Cre only. So we only get expression of this plasmid with Cre. So we get a simultaneous deletion of Baf53b in the mouse and then a reintroduction of the wild type. And then in our variant replacement condition, we have a similar flex on Baf53b plasmid, but instead I've introduced the variants of interest into the Baf53b protein. So we get deletion of endogenous Baf53b and then replacement with the variant. And so when I introduced these variants into excitatory neurons, I found a couple things. First off, with the 1177G to A mutation, I didn't actually see a reduction in branching. This was a little bit surprising, but I think it really highlights how it's important to do multiple mutations and to look at the functional outcomes of this. So you can see, once again, a nicely branched neuron that has the variant. And actually, if anything, it looks like at distal regions, the branches might be longer. However, this wasn't significant. When I did look at this C230A to G mutation, I did see a reduction in branching, and this is in that divergent region of Baf53b. I also created a plasmid with a complete deletion of that domain as a comparison. And you can see whether the domain is completely deleted or we introduce that point mutation, there's another severe reduction in branching similar to deletion, and that was significant as shown. So this really highlights that this region is important for the function of Baf53b in these neurons. And so this has all been in culture so far, and it's a great way to sort of investigate things quickly and initially, but what I really want to do is move this analysis into the mouse brain. And so to do this, I use a similar system where I have a Baf53b floxed mouse, and I can actually inject both of the Cre virus and the variant viruses into the ventricle of postnatal day 0 mice. And the virus will then circulate throughout the cerebrospinal fluid, and that will sort of infect the whole brain. And you can see that in this image of the brain here, showing the spread of the virus. And when we zoom in, we can see lots of neurons that have the Cre virus shown in red, as well as some neurons that also receive the variant virus. Those are the ones that are in yellow. And so I'm really excited to move forward with this and do some behavior on these mice to see if they have phenotypes of ASD, as well as to isolate these neurons out and do some more omics analyses on them. And so with that, I'll just conclude. I showed you that deletion of Baf53b stunts neuronal branching in both excitatory and inhibitory neuron cultures, and then replacing Baf53b with either the C230A-G mutation or a complete deletion of subdomain 2 results in reduced branching similar to Baf53b deletion. And so I have lots of future directions. I mean, this is a chromatin remodeling complex, and I'm still trying to figure out where it's binding in the genome in my system. So I really want to do cut and run for BRG1 and the different neuronal subtypes, particularly in the mouse brain. But I'm working on isolating out these neurons. It's a large organ with lots of cell types, and they're all interconnected. So this is a huge challenge in the field. And then again, chromatin remodeling complex, how is chromatin accessibility affected? And in turn, gene expression, is it important for neuron subtype function? I plan to do attack and RNA-seq in these neuron subtypes. And then lastly, do any of these Baf53b variants result in ASD-like behaviors in the mice? And could we potentially rescue these behaviors by reintroduction of Baf53b or another target that we might find once we do all of these analyses? And so with that, I'd like to thank the Cernia lab, Dr. Annie Cernia, a great mentor, and Marie, who has helped a lot with the inhibitory neuron project, and Fortune, who's been all around help on the whole thing, as well as sources of funding, and everyone for listening today. Thank you.
Thank you, Megan. That was fantastic. We already have one person with a couple people that have raised hands. So I'll ask Ben Weekly to unmute himself, and he can ask you his question.
Hi, Megan. Great talk. I had two quick questions. One was, did you notice or did you check if Baf53a was upregulated when you knocked down Baf53b? And then the second question, do you have any hypotheses about what this SB2 domain is doing, whether it's playing a structural role or functional role? Thanks.
Great questions. So I haven't looked at Baf53a, others have, and they do see that it's upregulated as well as still incorporated into the complex, but the complex can't do its job like it would in neurons. So it's not binding to the right places and it's not as stably associated with chromatin, but that's definitely something that I want to look at. And then the second question, the subdomain 2, I'm not sure. I mean, it's like a huge black box in the study of Baf53b. Potentially, it's mediating protein-protein interactions, potentially helping target Baf53b to regions in the genome, but it doesn't have a DNA binding domain. So again, we're all still trying to figure out what's going on with that.
Okay, so then next, Emily Brown has her hand up. So Emily, you can unmute yourself and ask your question.
Hi, Megan. Super cool work. The question that was just asked previously was actually my first question as well, looking at Baf53 expression. But I had a second question. I might have missed this. How did you select which mutation variants to screen? Were the two variants that you presented today, are those some of the most common mutations mapped in ASD?
Yeah, that's a great question. So most of the mutations are homozygous recessive, but I did select the 238 to G mutation because it's one of the dominant mutations. It's also in that subdomain 2. And then the 1177 G to A mutation is thought to be involved in protein-protein interactions with BRG1, which is the ATPase in the complex. So that was sort of the rationale there. They're also both present, or they're in individuals with ASD and ID. So it's potentially they're more severe, but I mean, I would love to look at all of them if I had the time and money, but that's sort of the rationale for why I chose those ones. There is another mutation, the 1027 G to A, which is a dominant mutation found in 10 patients, which we've also created the plasmid for, but I haven't actually infected that into the neurons yet.
And then next, Jackson Hoffman has his hand up. So Jackson, you can unmute yourself and ask your question.
Thanks, great talk. I'm curious if mutations in any of the other, or silencing any of the other BAF subunits causes a similar effect on branching. And do you have a hypothesis for how depletion of 53b might be disrupting the way the BAF complex interacts with the nucleosome?
Yeah, so that's a great question. So ARID1B, which is another subunit in the complex, shows similar things, as well as BRG1. Those are sort of the two most well-studied in neurons. And then how it's actually working here. So others have shown that it may be targeting the complex to certain areas of the genome, but the evidence for that is a little bit lacking. It was just basically like a chip experiment where when you knock down Baf53b, it's not going to the one region where they looked. And again, it doesn't have a DNA binding domain. So exactly how that's working, we're still not sure. And again, like when it's knocked out, it seems that the complex, although it forms and retains Baf53a, is not as stably associated with chromatin. So I'm looking into sort of those two avenues, but again, still lots of work to look at like protein-protein interactions and where it's actually going to the genome in neurons. There's also this whole other sort of a different area of study in the brain and specifically neurons, which I think Liz is going to talk about, where there's activity-dependent genes as well as like genes at baseline. And depending on what sort of system you're looking at and at what time point, it becomes really difficult to parse out whether they're activity dependent or not. So again, lots of things that I'd like to look at in the future, and I'm hoping soon to be able to isolate these neurons so we can actually look at this.
Okay, and then Gamal has their hand up. So again, I'll allow you to ask your question.
Can you hear me, Megan?
Yes.
Okay, thank you very much. Megan, I really enjoyed your talk,
Thank you.
I have a very general overview. First of all, in here we are assuming that this is specific, I would call it this gene, its expression, epigenetic expression, is related to autism. So my experience is as follows. Bardet-Biedl syndrome is a ciliopathy, and 90% of the children express autism in their majority. So it is associated that ciliopathies and the expression of the intrapharyngeal mechanism are defective in primary ciliary expression. So I would like to know if it is possible for you to carry out a genome-wide association study on Bardet-Biedl syndrome children. There must be plenty of genetic studies for these patients in some databases from the 1000 Genome Project, in which you can assess your hypothesis on these specific patients. Because my son, and this is the reason I am attending your presentation, your seminar, is because my son has Bardet-Biedl syndrome, and he expresses severe autism. He has the memory of an elephant. He can memorize everything from 50 years ago. But he has these specific issues. But he suffers from what we call is a ciliopathy.
Right.
And this is where I am coming from, is can you be able to test your hypothesis on these specific patients rather than animal models?
Yeah, I mean, you raise a really great point, and yes, we can. So others have done that with the Baf53b protein. So you can take like fibroblasts from individuals with ASD and differentiate them into neurons and look at them in culture. And so they've also seen defects in neuronal branching and some candidate genes that have come out. So these are things like Rho GTPases, things that lead to sort of downstream cascades that would be involved in neuron branching and synapse formation. But I think it's really important to highlight the fact that Baf53b is just one gene in this huge landscape of genes that are involved in ASD development. And there's actually not that many individuals that have been found with this variant. So that sort of schematic that I showed, those are each one is basically a patient because it's a recessive mutation, with the exception of those 10 dominant, the individuals with the 10 dominant mutations. And so what I'm really trying to do is look at the basic function of this chromatin remodeling complex, particularly Baf53b. And then maybe we can find something that converges with other genes that might be affecting ASD development as well.
So will it be possible to reverse engineer this process? Okay, so let's assume that you are able to obtain a biopsy from Bardet-Biedl children, and take it from the skin, and then say, I'm gonna reverse engineering and assess and have a comparative study with induced pluripotent stem cells, and see what are the deficiencies associated between normal, and do it on siblings, by the way. That's the most important thing. Do it on siblings. Because it's
absolutely doable. I'm not a clinician, but it's something that I would love to do. I'd love to partner with clinicians. There's a group in Montreal that actually has looked at these particular individuals. Scott Bell is the main author on that paper, if you want to look it up. And they've done some more of those sort of clinical things, but as of right now, I'm not partnered with any clinicians to look at. But I would love to do that work. I think it's really, really important for the community to look at actual individuals with ASD and to study their biology, right?
Awesome.
My
Thanks,
last-
Megan. Thanks,
Yeah,
Gabrielle. Sorry,
so-
we have to move on to the next seminar, so you can follow up offline.
Definitely contact me. We can talk more. Thank you.
Awesome. So thank you, Megan, for the wonderful talk and wonderful discussion afterwards. And I'll pass over to Sky to